So in this session, we are going to continue with Pentose phosphate pathway. In the previous session, we ended with the gluconeogenesis irreversible enzymes. And so if you haven't watched that video, you can go back and watch it because we need to build from the basics through the first aid until we finish. So that's the purpose of this series of review. If you haven't subscribed to the channel yet, it would just be nice to click on the subscribe button and click on the bell so that as soon as I post a video, you will be notified. Also, one reason why you should subscribe is that when you subscribe, then YouTube sees our video as relevant and then they suggest it to other people so they can also subscribe. So that will be helpful to me. Thank you for helping. If you're a returning subscriber, you are welcome. If you are a new subscriber, thanks for subscribing and join us so that we can all review this first aid together to make things easier for future students as well as present students. So we are beginning with the Pentose pathway, also known as the HMP pathway or the shunt. That is hexose monophosphate shunt. The purpose of this is to make NADPH. So it's making NADPH. Now, this NADPH is very, very important in the body. And it's from abundantly available glucose 6 phosphate. That is glucose 6 phosphate. Okay. Now, we all know glucose 6 phosphate. That is when glucose enters into the cell. The first step is to add a phosphate, which is the policeman, to the 6th carbon, the last carbon. That's why we call it the glucose 6 phosphate. So we are using that glucose 6 phosphate, which is here in this reaction, to make NADP. And that is the main purpose of the the Pentose pathway. Now, where do we use this NADP? We use it in several reactions, like in RBCs. We can use it to reduce glutathione so that glutathione can be used to destroy or reduce all the free radicals in the body or in the red blood cells. Now, when we have a lot of free radicals, as I said, free radicals are they are just toxic substances that are gotten from the end products of reactions. For example, when we have chemical reactions, certain compound like the superoxide, the O2 minus, like they just go freely and then they are toxic to the cells. When we breathe in fumes from the car exhausts, all those gases are free radicals, and then we need something like glutathione to remove them. So people use this glutathione as daily vitamins that they drink, and then as their medicine. So when they drink it, then it helps you to clear all the free radicals. One thing about the free radicals is that they increase aging. So if you Google on YouTube, somebody who is taking glutathione as a daily vitamin looks like younger than the time they were taking it. Like, yeah, it's really helpful. It also helps to some people use it to lighten the skin, and this way, like, the skin is not going to be like different colors, but it's gonna be brightened throughout since the drug is in the blood. Some also give this glutathione as infusions, and it's very helpful to them. Now, let's continue. So glutathione is reduced by NADPH. Usually, if you buy the glutathione, they've written on the bottle. If you look at it, they've written on the bottle, reduce glutathione. That's the best one. But the oxidized form is not active. It's less active. So we need NADPH to reduce it before we can use it to remove all the free radicals. Otherwise, we'll damage the red blood cell membrane and then we'll have hemolysis. The next place we use the NADPH is fatty acid and cholesterol synthesis. Very important. The most important one is the removal of the free radicals. Additionally, this pathway yields ribose for nucleotide synthesis. We'll see it down here. So let's just go straight. And then the phases are two phases. We have the oxidative phase, which is irreversible, that is here. And then we have the reversible phase, which is the non-oxidative phase. And then they love to ask questions on this, so pay close attention to this page when you reach it. So now we have glucose 6-phosphate. That's glucose. First of all, glucose coming to the cell, we add the phosphate by the glucokinase or the hexokinase, and then it becomes glucose 6-phosphate. Now if it becomes excess, we have like a lot of glucose 6-phosphate, we can use some of them for this pathway. So glucose 6-phosphate dehydrogenase, because it's coming to remove, this is glucose 6-phosphate, this dehydrogenase is going to remove a hydrogen from it. Then it will give the hydrogen to NADP plus so that it will become NADPH. Then the rest of the substrate will become 6-phosphogluconate. So this reaction is simple. So glucose 6-phosphate is converted to 6-phosphogluconate by glucose 6-phosphate dehydrogenase. How does it dehydrogenase? It removes the hydrogen from here and gives it to NADPH. Then NADPH will become it removes the hydrogen and give it to NADP plus. And then the NADP plus will become NADPH, like that. And then the rest will become glucose 6-phosphogluconate, and this will also be converted into ribulose 5-phosphate. And through this, so all this will occur. NADP plus will still will be reduced to NADPH, and carbon dioxide will come out. Then this ribulose 5-phosphate can be converted into ribose 5-phosphate. Then the ribose 5-phosphate, which is a ribose sugar, will be converted into nucleotide. That's DNA and nucleotide for DNA synthesis. That's the ATP, ADP, UDP, and co. So this reaction is irreversible. It cannot come back. But then we also have this reaction whereby the non-oxidative phase is done by fructose 6-phosphate. We know that glucose 6-phosphate, the next step is converted to fructose 6-phosphate. This fructose 6-phosphate can be converted into ribulose 5-phosphate using transketolase and B1. This is very high yield. This they call it the RBC transketolase. It's very, very high yield. Take note of this enzyme. If let's say somebody, or you meet a question that is talking about this pathway, and then we are using glucose to make ribulose 5-phosphate or to make nucleotide, you should also know that we can have transketolase also playing the same role. So if, for example, somebody has G6PD, okay, deficiency, and a child who has deficiency of this enzyme, but they still are able to make nucleotide. Why? Because they have transketolase that is working. But if both of them are absent, they cannot make nucleotides, right? They cannot make DNA and RNAs. Okay. Now, if we've seen that this reaction is what? Reversible, and so some of the questions love to ask about reversible reaction and irreversible reaction, and the most one that trick a lot of students is the transketolase reaction. So please pay attention to this. So these are the two enzymes that are very important, very, very important. These two enzymes are very, very, very important. So take note of this one here and take note of this one. Very, very important. Okay, now let's go to the next page. Now let's look at glucose 6-phosphate dehydrogenase deficiency, because here this pathway, the pentose pathway, is regulated by glucose 6-phosphate dehydrogenase. And then we said that glucose 6-phosphate dehydrogenase converts glucose 6-phosphate into ribulose 5-phosphate, generating NADP. Therefore, if you have deficiency of the glucose 6-phosphate, what do you think? This reaction will not occur. So you have reduced NADPH. What do we use NADPH for? We use it to reduce glutathione so that we can get rid of free radicals from the body. Therefore, if you lack glucose 6-phosphate, then you also lack what? If you lack G6PD, I'm sorry, if you lack G6PD, then you will also lack NADPH. Therefore, you can't remove free radicals. So those free radicals will damage the RBCs. So somebody with G6PD, this is the reason why their RBCs will be having hemolysis like that. So let's go quickly through this. G6PD deficiency. G6PD is necessary to keep glutathione reduced, which in turn detoxifies free radicals and peroxides. Decreased NADP in RBCs leads to hemolytic anemia. That's what we were saying earlier, due to poor RBC defense against what? Free radicals, which are the oxidizing agents. Those free radicals are the oxidizing agents. So it's the same thing. Now, because the free radicals, when they enter the cell, they oxidize the cell membrane and thereby damaging it. Examples of this are fava beans. This is high yield fava beans. So when somebody with G6PD eats fava beans, they can have hemolytic anemia. Sulfur drugs, nitrofurantoin. Nitrofurantoin is an antibiotic. We give it only to pregnant women because pregnant women have we give it only to pregnant women who have UTI because they are not it's not teratogenic. So just take note of that. If you are giving them, usually we give trimethoprim and sulfamethoxazole combined. That's Septrin. So if that one, that Septrin can block the DNA synthesis pathway, and you don't want to give that to a pregnant woman, that they'll get neural tube defect. You don't want to, so we rather give them nitrofurantoin. Primaquine is another thing that can cause increase in the free radicals, leading to hemolytic anemia if the person has G6PD. Others are the chloroquine and anti-tuberculosis drugs. Infections can also precipitate hemolytic anemia because infections also increase metabolism, and then metabolism will lead to increased free radical production. Now, it is X-linked recessive. It's an X-linked recessive disorder, and most common human enzyme deficiency, and most prevalent among descendants of populations in malaria endemic regions. That's sub-Saharan Africa and in the Southeast Asia. Histology on the RBC, you have Heinz bodies. That's denatured globin chain precipitates within the RBC due to oxidative stress. So in this, like when we reach it, you have to pause and look at how Heinz bodies look like. So the globin chains of the RBCs, they precipitate, and then that the hemoglobin, the globin part of the hemoglobin, they precipitate, and then they form like some solid thing inside the RBC, and that's all the Heinz bodies. Now, when these Heinz bodies in the RBC, when the RBC reaches the spleen, the spleen is going to remove the Heinz bodies, and then the cell will look like it has been bitten. That's the bites, or part of it has been bitten. So it results from phagocytic removal of the Heinz bodies by the splenic macrophages. Okay, so this is the pathway like this. So now glucose 6-phosphate, sorry, I don't know why all of them are highlighted. Okay, now let's continue. So glucose 6-phosphate was converted into 6-phosphogluconolactone by G6PD, by the enzyme G6PD, right? That's glucose 6-phosphate dehydrogenase. And how did it do it? It removed hydrogen from here, from this. It's G6PD because it's a dehydrogenase, so it's removing the hydrogen from this and then adding it to this to form NADP+. So glucose 6-phosphate removes hydrogen from the glucose 6-phosphate dehydrogenase remove hydrogen from glucose 6-phosphate and then add it to the NADP+ to form NADPH. Now NADPH has hydrogen, and addition of hydrogen to anything is called what? Reduction. So now NADPH will now stimulate what will be used by glutathione reductase to reduce glutathione. So originally glutathione is oxidized, and this is how it looks like, GSSG, That's the oxidized form. Now, if we add hydrogen, this hydrogen to it, it becomes this GSH. That's a reduced form. This is the active one. This is the part that the form that we can use to reduce all the free radicals, to remove the free radicals from the body. And then we'll add, for example, if we are adding glutathione to peroxides, so that we can remove the peroxides, it becomes the glutathione peroxidase. We use the glutathione peroxidase to do that, and then we'll convert, for example, hydrogen peroxide, because hydrogen peroxide, to water, because hydrogen peroxide is very toxic to the cells. And so glutathione peroxidase means this peroxide, right? Hydrogen peroxide. So peroxidase, we are breaking down the peroxide. We're using the peroxidase enzyme, and that is it. Okay. Now, I want to chip in something here that is not written, that I think is important. Is that now, if we want to know how much oxidative stress a cell is undergoing, okay, for example, the oxidative stress, I mean that how much of the free radicals are in the cell, we have to measure the ratio of GSS, the glutathione, the reduced form, to the reduced form to the oxidized form. And which of them do you think that will show that the cell has more, the cell is undergoing more oxidative damage? Increase in which of them? Is it the reduced form or the oxidized form that will show that the cell is being damaged by free radicals? Or another way I'll say that, which of them, okay, increase in which of them shows that the cell is under oxidative stress? Therefore, it is the GSS, because more of this cannot work, but this one reduces the free radicals. So increase in GSSG/GSH ratio, if that shows that the cell is undergoing oxidative stress. The same way, so I can write it this way. Let me put a text over here like this. So we can write it this way. If we have increased if you have increased GSS, G is to GSH. If we have increased this ratio, it means that the cell is what? There is oxidative stress, because this is the oxidized form. So this shows that there is oxidative stress. So equals increase more oxidative stress. Oxidative stress. Good, like that. So this is what you want to know, this, that if we have increased this, it means there is more oxidative stress. Also, we can also write it this way. Take note. It also, if we have decrease of this ratio, for example, if I write this way, if I have decreased GSH to GSH is to GSSG will also mean that there is more oxidative stress. So let's take note of these two things, which very, very important. Very, Very important. Okay, so decrease, if they bring this first, like GSH, it means that this one is more, the bottom one is more. That is why it is decreased. If we have decreased this ratio, it means the bottom one is more. This is the oxidized one, which this is the not good one. This one is not good. So if you have more of the not good one, then there is oxidative stress. Here also, if you have increase, it means that this one is less. That's why the ratio is increasing. So there's more oxidative stress. So to measure how stressful a cell is, we have to measure, if we want to measure how much oxidative stress is happening, we want to measure the ratio of GSS to GSH, and then we'll be able to determine that. Now, just something to chip in, very, very high yield. NBMEs love that. Disorders of fructose metabolism. So this is the disorders of fructose metabolism. And so yeah, so now we have two disorders of fructose metabolism. One is the essential one, and the other is the hereditary fructose intolerance. It involves defect. The essential one, it involves the defect in the fructokinase, enzyme. If you remember, it's the same, like just like we have glucokinase. When glucose enters the cell, we don't want it to go back out, so we use glucokinase or hexokinase. The same way, if fructose enters the cell, we use the fructokinase to pin it into the cell so that it cannot go out. It's autosomal recessive. Most of the enzymes, majority, like 80% of enzyme deficiencies are autosomal recessive. So just take note. If you don't know and it's an enzyme deficiency, it's autosomal recessive. Just few of them are not. It's benign and asymptomatic. That's why they say fructokinase is kinder. Since fructokinase is not trapped in the cell, hexokinase becomes the primary source of converting fructose to fructose phosphate. So we know that hexokinase can work on all monosaccharides. Hexokinase can work on glucose, fructose, and galactose, but glucokinase can only work on glucose, right? So therefore, if we don't have a fructokinase over there, hexokinase can still work on fructokinase. That is why its deficiency will not really lead to any major problem. So symptoms are fructose appears in the blood and the urine. So there'll be increased fructose in blood and urine. So they'll give you the question, and then they'll say there is increased fructose in the person's urine, or they'll tell you there's increased or reducing a substance in the person's urine. Is the same thing. Disorders of fructose metabolism cause milder symptoms, so that's why they say it's kinder than the other it's analogous disorders of the galactokinase. Galactokinase is not it's not kind like fructokinase. Now, the second one is hereditary fructose intolerance. So in these people, they have deficiency of aldose B. Aldose B. You remember in glycolysis, aldose B was the one that was splitting the fructose 1,6 base phosphate, right? It split it into two. So that was aldose B. So here, aldose B also splits fructose 1-phosphate into dihydroxyastone phosphate, that's DHAP, and then glyceraldehyde. The same as in glycolysis. So deficiency of aldose B will lead to accumulation of fructose 1-phosphate. And so let's read. Fructose 1-phosphate accumulates, causing a decrease in available phosphates, because the phosphates are trapped in the fructose 1-phosphate, resulting in inhibition of glycolysis, because glycolysis needs phosphate. Gluconeogenesis also needs phosphate. So symptoms present following consumption of fruit, juice, and honey. and urine dipstick will be negative. So negative test for glucose only. Now, reducing sugars can be detected in the urine. So there'll be reducing sugars in the urine, but then it will be those reducing sugars will be negative for glucose. So if they tell you they are reducing sugars in the urine, but it is not glucose, then you are thinking of either fructose or galactose. Most likely it's going to be fructose in the urine. Symptoms are hypoglycemia, jaundice, cirrhosis, and vomiting. So one way I appreciate this is that when I see the one over here, I feel like the one is like a pin that is going to prick the liver. So if I take fructose 1-phosphate, if I take accumulation of fructose 1-phosphate, it's going to prick the liver. So it will cause liver problem, right? Now, if I also take galactose 1-phosphate accumulation, it's also going to cause pricking of the liver. So anywhere you see 1-phosphate, fructose 1-phosphate, galactose 1-phosphate, liver problem will be there. That's how I remember it. Now, treatment is decrease in fructose. So of course, if somebody cannot break down fructose, why do you give them fructose? You don't give them. You also reduce sucrose, because sucrose is made from glucose and fructose put together. Therefore, if you give them sucrose, which is the common sugar that we eat, the table sugar, and they'll still have fructose from it, which is not very good. Sorbitol should also be reduced because it's metabolized to fructose. Sorbitol can be converted into fructose so that to be metabolized. That's how we break down sorbitol. Now, so this is the pathway. Fructose normally converted to fructose 1-phosphate by fructokinase. If it's deficient, we'll have excess fructose building up. Is that correct? Yeah. So we'll have excess fructose building up. This fructose will be seen in the urine, and it does not really cause any symptoms. That's the kind one. Fructokinase is kinder. Next is aldose B. Aldose B deficiency leads to accumulation of fructose 1-phosphate, which is the hereditary fructose intolerance. And then they'll have accumulation of fructose 1-phosphate, which will prick the liver and cause liver problems. Now, let's look at disorders of galactose metabolism. So metabolism of galactose, one of them is galactokinase deficiency. The second one is classic galactosemia. Let's look at the difference. Galactokinase deficiency is hereditary deficiency of galactokinase, and galactitol will accumulate. So let's look at the reaction. Now, this galactose, it has entered the cell, right? What do? We use galactokinase to convert it into galactose 1-phosphate because we don't want it to leave the cell. Therefore, we have to add a phosphate. So we have galactose 1-phosphate. Now, galactose can also be converted into galactitol by aldose reductase. So if we do not have, or we are deficient of galactokinase, what will happen? This reaction will move in this direction and we'll have a lot of galactitol in the body, right? That is the result of galactokinase deficiency. So we have increased galactitol caused by aldose reductase. And galactitol accumulates if the galactose is present in the diet. Relatively, it's also mild condition and autosomal recessive, as we said. Symptoms are galactose appears in the blood and urine, just like fructokinase deficiency. And infantile cataract. So here they have cataract, but remember that fructokinase deficiency, they don't have cataract. Okay. Fructokinase deficiency, there is no cataract, but galactokinase deficiency, they have cataract. So remember that. Very, very important. Very high yield, they have cataract. Very high yield, they have cataract. So take note of that. Now, so here you see they said galactokinase deficiency is also kind. It's also kind. So this versus this, galactokinase is kinder than the classic galactosemia. The same way fructokinase is also kinder than the hereditary fructose intolerance. Now, the next one is the classic galactosemia. This is the main one. And I told you that anytime you see one phosphate in the thing, the one is going to prick the liver, right? So this one is caused by deficiency of this enzyme here. That is absence of galactose 1-phosphate uridyl transferase is autosomal recessive. So all of them are autosomal recessive. Damage is caused by accumulation of toxic substances, including galactitol, which accumulates in the lens of the eye. Symptoms develop when infants begin feeding. That's lactose present in the milk. So as soon as they begin feeding, they can have this problem. And then routine formula and includes failure to thrive, jaundice, and hepatomegaly, and infantile cataract. Intellectual disability. They can also predispose to E. coli sepsis. This is very important. This is very important because a lot of people don't pay attention to it. So you would like to ask it. So classic galactosemia can present with E. coli neonatal sepsis. And here, the high yield things that you want to know is that galactose 1-phosphate will cause hepatomegaly, right? Because the one over there will prick the liver, and then that will cause liver problems. So hepatomegaly, take note. Once you see that the liver enzymes are high, maybe AST, ALT are high, or they tell you the person has hepatomegaly, you want to start looking at the classic galactosemia, or this one here, hereditary fructose intolerance. But most likely the galactosemia is the one that causes the hepatomegaly. Or cataract is also in the classic galactosemia, and it's also in the galactokinase deficiency, but it is not in the essential fructose urea, because we said hexokinase can still work. Okay, so cataract is down here. It's not in the first two, but it's in the galactose side. Okay, and I don't know if you know the mnemonic galactic crab. Okay, that one is in the crabby, galactic crab. Okay, came to destroy my eye. So this is it. So if we want to look at the pathway again, galactose converted into galactose 1-phosphate by galactokinase. Deficiency will lead to accumulation of galactitol. Uridyl transferase, that's galactose 1-phosphate uridyl transferase. What does that mean? It means that this enzyme, okay, is transferring uridyl to galactose 1-phosphate, just as we see here, transferring the uridyl to galactose 1-phosphate. So the mnemonic is FabGat. I didn't use this mnemonic myself, but my study partner used it. Fructose is to aldose B, as galactose is to uridyl transferase. FabGat. That's a nice mnemonic. Next, sorbitol. This is highly tested. An alternate method of trapping glucose in the cell is to convert it to an alcohol counterpart, which is called the sorbitol. The OH at the end of it, the functional group that we talked about, the O makes it an alcohol via the aldose reductase pathway. Some tissues then convert the sorbitol into fructose. Very, very high yield. This information, conversion of sorbitol into fructose. Now, this means that we know that when sorbitol accumulates in any cell, sorbitol is hydrophilic, so it draws a lot of water onto itself. For example, if somebody has diabetes, and then in diabetes they have excess sugar in their body, right? So the sugar will be converted into sorbitol. Now, if the sorbitol accumulates in the eye lens, for instance, in the lens of the eye, you realize that the sorbitol start drawing a lot of water into the eye, and that will cause osmotic damage to the lens, for instance. So if the sorbitol is not able to be broken down. If we can't break down the sorbitol, then it will draw water. But if we can break it down, then it's not going to draw any water. So they want us to know that certain tissues in the body have what it takes to break down the sorbitol. That is the sorbitol dehydrogenase. So sorbitol dehydrogenase can also break down sorbitol. Now, not all tissues have it. The tissues that have it are, so let's read, the liver, ovary, and seminiferous vesicles, okay, or seminiferous tubules have both enzymes. So they have the aldose reductase and also the sorbitol dehydrogenase. So these, the liver, the ovaries, the seminiferous tubules cannot have sorbitol accumulate to cause osmotic damage, right? So they have both enzymes, so they are good, right? They are good. They lose sorbitol, so they are the best. They are the good guys. Now let's look at the lacks. They lack, right? The lens, the eye lens. That's why diabetics can have retinopathy easily, because the sorbitol will accumulate and draw water into it. Now they cannot see very well. Retina, so the lens, the retina, the kidney, and then the Schwann cells. That's why they can also have neuropathy, because the Schwann cells will accumulate sorbitol and then they cannot break it down. So it will draw more water into our nerves, which is not good. So LAX, that's a mnemonic: the lens, the retina, the kidney, and then the Schwann cells. Very important. So LAX, lack sorbitol dehydrogenase. LAX, the LAX lack sorbitol dehydrogenase. Yeah. Now, I'm sorry about that. So do not disturb. Okay. Let's go to lactase deficiency. What is lactase deficiency? It's an enzyme that breaks down lactose, right? So lactose is in the milk. It's mostly in milk. So if lactose is in milk and then we have lactase deficiency, means that we cannot break down the lactose. And what will happen if we can't break down lactose? It will cause diarrhea, right? So let's just go through it quickly. Insufficient lactase enzyme leads to dietary lactose intolerance. So anytime the person eats milk and milk contains lactose, they cannot. So the lactose will accumulate in the GI tract and pull water from the GI tract to itself, and that will cause osmotic diarrhea. Lactase functions in the intestinal brush border to digest lactose that is in the milk. So, and then we break down the lactose into glucose and galactose, as written here. Now, primary. So we have the primary form and then the secondary, and also we have the congenital one. So primary, so that's age-dependent decline in after childhood. So as we grow, it reduces because children eat milk, so they have a lot of lactase in them. But as we grow, it reduces. That's why adults who drink a lot of milk, they need to drink more milk to be able to get more the nutrients from it. And now the secondary one is loss of the intestinal brush border due to gastroenteritis. So after infection, especially the rotavirus or autoimmune diseases, then we lose the lactase. It's one of the common. So to present this way, somebody who had maybe gastroenteritis or rotavirus infection some months ago, and then now start having diarrhea, or can be due to a secondary lactase deficiency. We also have the congenital one, whereby the person was born with a deficiency, and then this due to a genetic defect. The stool demonstrates decreased pH. Decreased pH means that the stool is acidic, and the breath shows increase in the hydrogen content with lactose hydrogenase breath test. Lactose hydrogen breath test. H plus is produced when the colonic bacteria ferments the undigested lactose, right? So that reduces the pH. So just take note. when lactase is not there to break down the lactose, the bacteria in the colon is going to break down the lactose, and that will release a lot of H plus. This H plus is acid, right? So it will make the pH decrease, and that is it. So intestinal biopsy reveals the normal mucosa. Let's take note of that. Okay, versus the celiac disease. Versus celiac disease, whereby there's blunting of the intestinal brush border. This one, which is the lactose deficiency, there's no blunting of the intestinal brush border or the villus. Now, the findings are bloating, the abdominal bloating, cramp, flatulence, since we said the colonic bacteria will act on and ferment the lactose, and then also they have osmotic diarrhea, especially after eating milk. Treatment: avoid dairy products, so avoid milk or lactose-free diets. Now, this is another interesting subject here. So this is important to memorize. Do not skip it. I know it's not interesting, and many students are likely to skip it, but do not. Okay. I have a mnemonic. I don't have my book here. Where's this book? I wish I had a book here. Okay. Now, if I don't see it, let's continue. Amino acids. So these are the summary of the amino acids. So only L amino acids are found in proteins. So let's take note of that. Only L amino acids are found in proteins. So there are essential ones and the non-essential ones. The essential ones are what we must eat from our diet. The non-essential ones can be made from the body. Amino acids can be divided into different forms. We have the acidic ones, the basic ones. We have hydrophilic ones, hydrophobic ones. Right, we have those that function in different things. So we want to memorize this in any way that we can, because NBME love asking questions on it. So you want to understand this, okay, at least in some way. So we have private team hall. This private team hall is all the essential amino acids. Private team hall is all private team hall. They are all the essential amino acids, and these are their names. These are mnemonic: phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, leucine, and lysine. They are all essential. We need them from our diet. Now, we have the glucogenic ones, those that can be used to make glucose. The methionine, histidine, valine, and they said what? We met his Valentine, who is so sweet. That's glucose. So we met his, that is, met his, we met his Valentine. Met his Valentine. So Valentine is sweet. So met, methionine, or methionine, histidine, and valine. We have those that can be used to make both glucose and ketones, so glucogenic and ketogenic. That is isoleucine, phenylalanine, threonine, and tryptophan. We have those that are only ketogenic. That is leucine and lysine there. And we have the basic ones, which are arginine, histidine, and lysine. Arginine is the most basic one. Histidine has no charge at body pH. Therefore, histidine has no charge at body pH. So we will see that there is an for example, when we have okay, let's continue. I'll bring that concept later. So the acidic ones are aspartate, as we said, the names are the aspartic acid, right? And then glutamic acid. Their names even suggested that they are acidic. So they are very simple: glutamic acid and aspartic acid. They are the acidic ones. So his lies are basic, is a mnemonic for the basic amino acid. They are the basic ones. But then in the body, histidine, even though it's basic, but it's not going to be basic at body pH. So my mnemonic is like this. Let me see. just for people who want it. It's like pro, like this, pro gav peel fierce water. That's my mnemonic, fierce water. Then the next is veil has a branch. Then the next is his lies are basic. His lies are basic. Or let's write it his lies. Why? His lies are basic. And then the last one, because we are going to sing this into a song so that you will remember. That's how I remembered it. And then L and L ketones. These are the ketogenic ones. So this is how I remember my mnemonic. Yeah. Good. It's like this. Good. So I'm just gonna sing it, and then what I do is that like I try to sing this over and over and over and over, and it sticks in your mind forever. Okay, so that you can use it to answer these kind of questions easily. There are easy points that you can get on any exam. On my real deal, I met some questions from here. NBMS, I met questions. UWorld, I met questions. So it helps me. So pro gav peel. So this is proline, and the G is which one begins with G? Glutamine, arginine, right? Valine. Another P, phenylalanine, isoleucine, and leucine. So these are the hydrophobic ones. They fear water. So pro gav peel fears water, like that. Then veal are the branch chain ones. In what is it? Maple syrup syndrome, whereby we have the branch chain ones. So veal has a branch, right? Veal has a branch. That's the maple syrup. So when, for example, when the child has their urine smells like sugar or something, then that's the maple syrup. Okay. Now the next one is the his lies are so basic. His lies are so basic. Right? Basic lies. Okay. Then L and L ketone, leucine and what lysine are used to make ketone. So I'm going to sing it for you, and then you learn it from there. So pro gav peel fears water, veal has a branch, his lies are so basic, L and L ketone. This is my own mnemonic. I formed it myself. Copyright. I have copyright over it. Okay, so yeah, it's like this. So pro gav peel fears water, veal has a branch, his lies are so basic, L and L ketones. And so this helps us to memorize this whole thing. We know that the ketogenic ones are this, the basic ones are this. We'll see why like the importance of their basicity. And then we also have the veal, which is a valine, leucine, and isoleucine have a branch, because they are the branch whereby we have deficiency of the branch chain ketoacid dehydrogenase, and then we'll have the maple syrup syndrome, right? Okay. And now here we have his lies are basic. Now why are these important to us? Because they are used in the DNA synthesis, whereby a DNA, since the nucleic acids are what? They are acids, as the name suggests, nucleic acids. They are acids, so they need some basic thing to wind against, right? they need some basic thing to wind again. So the histidine and lysine are important in that sense. Okay, let's go to the next thing. Another high yield, very very high yield. You will not meet an exam without this. Like, then it's not an exam. Okay, urea cycle. They want us to understand what is urea. Okay, urea is the end product of what? Metabolism of proteins, right? Okay, let's see how we form it. So amino acid metabolism. So we break down proteins into amino acids because we build proteins with amino acids. So we break proteins down into amino acids, right? And then so amino acid catabolism generates common metabolites. That's pyruvate, acetyl-CoA, which serve as metabolic fuels. Excess in nitrogens is converted into urea, right? Because we have a lot of nitrogen in the amino acids. So as we said, because we said the functional group is NH2. So that nitrogen that was in it, we need it, the NH2. Good. The functional group is NH2. So this NH2, after breaking down the amino acid, we need we'll have to do something with the NH2. So the nitrogen part is what is going to form the urea at the end of the day. Now, what do? We are going to combine carbon dioxide with ammonia in the body, and that will form carbamyl phosphate. Now, this carbamyl phosphate is being synthesized by an enzyme called the carbamyl phosphate synthase 1. There are two. This one is the synthase 1. The 2 is found in the cytosol, but the 1 is found in the mitochondria. So 2 for cytosol. So cytosol for 2. Now we use ATP in this process. So ammonia plus carbon dioxide forms carbamyl phosphate using carbamyl phosphate carbamyl phosphate synthase 1. Now this carbamyl phosphate will now be added to ornithine to form citrulline. And what does that is the ornithine transcarbamylase. What does that mean? It means it is transferring carbamyl phosphate to ornithine. Ornithine transcarbamylase. It transfers carbamyl phosphate to ornithine to form citrulline. Now citrulline goes through this process. So from citrulline we get adenosine using adenosine synthesis. Then we get what? We get arginine, but in the process we give off fumarate. This fumarate will go into the TCA cycle. We saw it earlier. That's linked to the TCA cycle. And now arginine will be converted into what? Ornithine again. So it's a cycle using the arginines, but here we give off the urea, which goes into the kidney to be eliminated. So this side is occurring in the cytosol. This side is occurring in the mitochondria. So this is it. But the pathway is not the main thing. The main thing here is that you want to know that when there is a deficiency, what would the problem be? So in this reaction, the most common deficiency is the ornithine transcarbamylase. That's the most common one. This one here is the most common one. If you don't know the answer, it's most likely to be the answer. Ornithine transcarbamylase is most likely to be the answer. So take note of it. It's most likely to be the answer. All right. Now, let's look at how it will look like. So when you have ornithine transcarbamylase deficiency, what do you think will happen? We will not be able to form citrulline. So we'll have decreased citrulline. So citrulline will decrease. Is that correct? Yes. So we have low levels of citrulline, and also we'll have what? Increased ornithine, because we cannot convert ornithine. Next, we'll have increased what? Carbamyl phosphate. right? And then we'll have increased ammonia because everything will back up increased ammonia. So this is highly tested, so you want to really take note of it. Now, you don't just learn this in isolation. The NBME or the board, they want you to be able to differentiate between three things. I'm going to type them here. If you can differentiate between these three things, then you are good. Then you will always get the answer correct, and it will increase your scores. So ornithine transcarbamylase, OTC, I'll call it OTC, deficiency, ornithine transcarbamylase, OTC. The next one is, so let's just write what is there. In this one, we will have low levels of citrulline. We have low levels of citrulline, as we said earlier. Take time to memorize this very well, okay? Because it will help. You will always get the answers correct. Low levels of citrulline. You want to get this, you'll get high ammonia, right? Because everything will back up and then we'll have high ammonia. Good. So I'm going to write high ammonia. High ammonia. Then the next one is that we'll have high orotic acid. Okay, high orotic acid. Now somebody may ask, where from the orotic acid? If you want to understand this, then we have to go to page 36. 36, just hoping that is it, where we have the the pyrimidine and purine synthesis pathway. So I'm going to search for it over here, and then we look for it. Page 36. Page 36. This is it. Good. Now, what did we say? We said we are talking about the ornithine transcarbamylase deficiency, and then we have high orotic acid. So if we do not have the ornithine transcarbamylase, what is happening is that the carbamyl phosphate that was going to form, the carbamyl phosphate that was going to accumulate, that carbamyl phosphate will now come into this pathway. The pyrimidine pathway will be used in the pyrimidine pathway, because we said carbamyl phosphate will increase. That carbamyl phosphate will come here and then be converted into this orotic acid. And that is why they said orotic acid would be increased. So please take note. Please take note. Okay, so the orotic acid will be increased. That is why it's like that. Let's go back to our page. See, that was page probably 70. This is page 80 probably. Not very sure, but if we get to page 80, we can maneuver away from there. Okay. All right, so we're somewhere here. So let's go back to where we were. Good, we were here. So the carbamyl phosphate that is going to accumulate will go into the pyrimidine pathway and then be converted into orotic acid. So we will have high levels of orotic acid in the deficiency. Now, the next thing that you want to differentiate it from is the carbamyl phosphate deficiency itself. carbamyl phosphate deficiency. So I'll call it CPD, carbamyl CPS deficiency. Yeah. So CPS deficiency, CPS, the carbamyl phosphate synthase or synthesis deficiency. In this also, we will see, of course, if you look here, what is happening? If you have this deficiency, we'll have increased ammonia. Is that correct? Yeah, we'll have increased ammonia. We'll have low carbamyl phosphate. Is that correct? We'll have low carbamyl phosphate. If we have low carbamyl phosphate, that means we'll have low orotic acid. Is that correct? So first of all, let's say increase high ammonia. So these things are simple. You just have to understand the pathway. High ammonia. Sorry. I don't. High ammonia. Then we again said what? If we have high ammonia, previously I showed you something that carbamyl phosphate can be used to make orotic acid. So we'll have low orotic acid. So this is how the boards, they'll present the questions to you. They'll give you all these things and tell you, oh, the person has high this, low that, high this, low that. And you have to be able to picture and deduce it. Also, so we said high ammonia, low orotic acid, will have low carbamyl phosphate because this is deficient. That one just makes sense. So low carbamyl phosphate, low carbamyl phosphate. And what again shall we have? We also have megaloblastic anemia, because if you saw the pathway that I showed you, the pyrimidine pathway, we are using it to make pyrimidines, which will make ATP, and then at the end of the day, they will make nucleic acids, which are DNA and RNA. So if we do not have that, then we'll have megaloblastic anemia. Megaloblastic anemia. And what you want to know is that in this megaloblastic anemia, if we treat with, it's not B12. So if you treat with B12, the person will never get well. The person will never get better. So it is resistant to B12. So take note of that, to resistant to B12. To B12, vitamin B12. Okay. Now, the next enzyme, deficiency that we want to compare this is the orotic aciduria itself. So orotic aciduria itself. So that's the third one that we want to compare it to. So if you know these things, then you are good. So in orotic aciduria, if you want to remember it, let's see. Let's go back to page, let me see, page 36. It will not show us. My watch is making noise. I'm taking my time so that people can really understand and appreciate this. So if you feel the video is, if you know these pages, you can increase the speed. But if you don't understand, just take your time and then we will do it. This is how we all did it. So let's go to page 36 so that we see orotic aciduria itself. It's loading. So in page 36, what is happening? Okay. Good. So in page 36, you realize that now we are talking of orotic aciduria, meaning that we have increased orotic acid in the urine, right? So where does this occur? It is, we use orotic acid to make UMP. But what enzyme do we use? It is UMP synthase, right? So the deficiency is UMP synthase deficiency. So let's take note of that UMP synthase deficiency. What are we going to have? We are going to have ammonia to be normal, because this does not affect the ammonia pathway in any way. So we'll have ammonia to be normal, and we'll have increased orotic acid in the person's urine. So let's take note of that. So let's go back to our page, maybe 83 or so. Page 83. Or it's 82. Okay. So we were here. So this one is equal to UMP synthase deficiency. UMP synthase deficiency. Okay, and then here we will have normal ammonia. So it looks like this is the only one that has a normal ammonia. All of them have high ammonia. Only this one has normal ammonia. So if you have normal ammonia, it's most likely to be right. So you have to differentiate these three things. If you can, you always score a point on the NBMEs. So thanks. Now let's go to the next one. The next page says transport of ammonia by alanine. So here we can see it starts from here and it finishes here. So amino acids here, okay, and this occurring in the muscles, this occurring in the liver. So all they are trying to say that alanine transports ammonia, or alanine can transport ammonia. Yes, alanine can transport ammonia, of course. Alanine is an amino acid, so I'll not be surprised if it's transferring ammonia, because it has NH2 already, so it just needs one hydrogen to form the ammonia. Now, amino acids, what they are doing is that here it's giving NH3 to alpha ketoglutarate, right? And then it becomes the skeleton will be the alpha keto acid skeleton, and now here will form glutamine. So amino acids give ammonia to alpha ketoglutamine glutarate to form glutamate. Now glutamate will now transfer the NH3 to pyruvate in order to form alanine and alpha ketoglutarate again. So this will be a cycle. Now from the muscles through the Cahill cycle, alanine can transfer ammonia to the liver. Alanine will move from the muscles to the liver, right, through the Cahill cycle, like this. In the Cahill cycle, you realize that pyruvate takes this and takes the ammonia and gives, and then like pyruvate takes this ammonia and then it converts it to alanine and then alpha ketoglutarate. Then the alanine can now move out of the cell and circulate into the liver. So alanine can move out of the cell. That's what we call it, the alanine shuttle. Can move out of the cell, and it's holding that ammonia that we got from the original amino acid here. And now this alanine in the liver can also transfer the ammonia back to the alanine can now break down and then transfer the ammonia back to alpha ketoglutarate again, just like here, to form pyruvate and then a glutamate. And now this glutamate will now have the ammonia that can be used can be broken down in the liver to form urea, like that. Yeah. So all we are trying to do is we are using the ammonia to form urea, and that is it. It's not really high yield. Hyperammonemia. What does that mean? We have excess ammonia in our body. This can be acquired by liver disease, mostly when someone has liver cirrhosis or their liver is not there, anything that causes severe liver disease, because the liver is supposed to remove all the ammonia from our body and then metabolize them into urea. So if that cannot happen, we'll have hyperammonemia. And they present with flapping tremors, hysteresis, and then slurred speech. So they'll be there and their hands will be shaking, flapping. And then they have somnolence, like they are very sleepy because of encephalopathy. And then they have vomiting, cerebral edema, and also blurred vision. So increased NH3, and that's ammonia, changes relative amounts of alpha ketoglutarate, glutamate, and GABA and glutamine in favor of increased glutamine. So CNS toxicity may involve decreased GABA and decreased alpha ketoglutarate. Now, TCA cycle inhibition and cerebral edema due to glutamine-induced osmotic shift. So, some resources really explain this well. What they want you to know is that when we have excess ammonia, for example, in the GI tract, okay, the ammonia will be absorbed into the blood, and now it will combine with glutamate, and then when it combines with glutamate to form glutamine. Now, excess glutamine can draw water. Excess glutamine, which will be in the neurons, will draw a lot of water into the neurons, right? Another way is that excess glutamine can also activate calcium. Okay, it will activate the calcium, because we know calcium can cause excitotoxicity. So glutamine and calcium can cause excitotoxicity. So excess glutamine will cause excitotoxicity, meaning that it will damage the neurons, and like that, by pulling a lot of water to itself. And when it pulls water to itself, the person will have cerebral edema, because all the brain is full of neurons and water. Now, treatment is we limit proteins. So somebody with liver cirrhosis, we want to limit proteins, also have excess ammonia, and then they'll start having cerebral edema, and then they'll start having confusion. And maybe given to decrease the ammonia. So we can give lactulose. So these things may be given to decrease the ammonia. So lactulose acidifies the GI tract, right? And then traps the NH3, which is ammonia, into NH4, because acidifying the GI tract means that we are forming H plus. So H plus the ammonia that is excess, which is the NH3, will form NH4, which is the ammonium, right? This ammonium, okay, for excretion. This ammonium will be excreted. Antibiotics, we can give rifaximin, very high yield, very high yield. Rifaximin is very high yield, so you should know it. And then neomycin, and then we give these antibiotics so that they destroy the GI bacteria, and then the GI bacteria cannot convert. They can't convert the proteins in the GI tract. They can't convert it into NH3. Now, benzoate, phenyl acetate, and phenylbutyrate react with glycine and glutamine, forming products that are excreted renally. This is not a high yield information. Let's go to the next one. So here they are treating the ornithine transcarbamylase deficiency for us, and we've done it already, so we are happy. We just go through it. So most common urea cycle disorder is X-linked recessive versus urea cycle enzyme deficiency, which is autosomal recessive. It interferes with the body's ability to eliminate ammonia, often evident in the first few days of life, but may present later. Excess carbamyl phosphate is converted into orotic acid in the pyrimidine synthesis pathway. So whatever we said is the same thing that they are saying. The findings are increased orotic acid in the blood and urine, right? Because we said the carbamyl phosphate will be used to make orotic acid, so we have increased orotic acid. And then we'll also in the urine as well, and we'll also have decreased BUN, because the urea that would have been forming in this cycle will not be formed, so BUN will be decreased. It's the same as decreased urea, because urea and nitrogens, right? This is just blood. Urea and nitrogen in the blood. The symptoms of hyperammonemia will also be same, because we'll have excess ammonia, and some of the symptoms are confusion and hysteresis. No megaloblastic anemia versus orotic aciduria. Orotic aciduria has orotic aciduria will also have megaloblastic anemia because we have the UMP synthase deficiency. So we've truncated that. So we'll also have megaloblastic anemia in the orotic aciduria. So let's add it here. If we didn't add it earlier, orotic aciduria, the normal ammonia, and here we'll have megaloblastic. So anything that affects the pyrimidine pathway has megaloblastic anemia. So megaloblastic anemia. Okay. And of course, this megaloblastic anemia will not be cannot be treated with B12. So it's B12 resistant. Yeah. Now, derivatives of amino acids. After this, we will end it here and then we'll start this because this is another very high yield. I got a question from this in my okay. Now, amino acid derivatives. So these are the things that we use amino acids to make. Now, phenylalanine, this chapter is high yield. Please take it serious. Memorize every single, play with them. You will know them. Phenylalanine can be used to make tyrosine. Then the tyrosine can be used to make dopa. And dopa can be used to make dopamine. Dopamine to norepinephrine. Norepinephrine to epinephrine. What you want to know is that these things that are up here, you want to know them. The BH4, B6, vitamin C. This vitamin C, gosh, this one here, this is the one that beats me. Should have known better. Okay, now phenylalanine, because it came in my something. It came in my something, then I didn't get it correct. It hurt my feeling up to today. Okay, now phenylalanine is converted to thyroxine using BH4. Now, BH4 also mediates conversion of the thyroxine to dopa. Now, when we come down here, we'll see the enzymes. So we'll do this another day, but we just have to know this. Now, tyrosine can be used to make the thyroxine, and also dopa can be used to make melanin, the melanin that makes us dark. And tryptophan is used to make niacin, that is vitamin B3, and also tryptophan can be used to make serotonin. Therefore, if somebody has serotonin syndrome, they'll have excess serotonin, right? And all this means that the pathway will shunt to this way, and the person will have vitamin B3 deficiency. So people who have serotonin syndrome have vitamin B3 deficiency. Take note. Now, niacin, we also said it is what I call it. Another name is the NADH or NADPH, like that. So niacin can be used to make NADH, nicotinamide adenine dinucleotide phosphate. Now, also remember that serotonin can be used to make melatonin. Serotonin will keep us awake, and melatonin will make us sleep. Histidine can be used to make histamine. This one, the name suggests. Now, glycine can be used to make heme. Glycine and succinyl-CoA can be used to make heme. So take note of that. So glycine can be used to make porphyrin to heme. Glutamate can be used to make GABA and glutathione. Glutamate, glutathione. Also, glutamate can also be used to make glucose. It's also glucogenic, right? Now, arginine can be used to make creatine, urea, and nitric oxide. Very, very high yield. This nitric oxide one, very high yield. Very, very high yield. So take note of it. So BH4 is called tetrahydrobiopterin. Yeah, so we'll end this video here, and then in the next video we'll continue from the catecholamines and we'll continue with all the deficiencies. So thanks for watching this video. Share with your friends, let them join us for this classic video, this classic review of the USMLE First Aid, and If you haven't subscribed yet, do subscribe to our channel and also share with your friend. Do comment where you think we could have done better. Let us know, because it all helps in our improvement, and your questions are also welcome. Thank you and good luck in your studies. You can do it. This is how we all did it. You can do it. Believe in yourself. Peace out.